I'm Irfan, and today I'll present my project on cloning an efficient expression of human insulin in E. coli using PET28A plus vector. So the aim is to produce insulin efficiently in a bacterial system, so it could be easily purified using the N-terminal histag in the vector. Now I want to explain what the insulin is. Insulin is a hormone that regulates the blood glucose and essential for treating diabetes. The coding sequence is 333 base pairs for RefSig, and starting with the codon ATG and ending in the codon TAG. So, this diagram summarizes the process from selecting the insulin gene, cloning into PEN28A plus, and actually transforming E. coli BL21, inducing expression and purifying histag protein. Say, why I chose PET28 plus vector? I chose this because it is a strong T7 promoter for high expression in BL21, which can be induced with IPTG, and it also has the N-terminal histag for nickel-based purification, a thrombin cleavage site, multiple cloning site where insert insulin, and a chlamycin-resistant gene for bacterial selection. And the restriction sites that I use are NDI at 5' end and ZOI at the 3' end. So I want to talk about primer design. I designed my forward primer with the GC clamp, the NDI site CATATG, and the first 20 nucleotides of the insulin coding sequence. And my reverse primer as a GC clamp, the ZOI site CTCGAG, and the reverse complement of the last 20 nucleotides, including the tag subcodon. So the protein terminates naturally with only the N-terminal histag. I'm using two different restriction sites. Ensures that the insert is cloned directionally and avoids self-ligation. First, I taught the competent E. coli cells on ice and gently mixed them with my recombinant plasmid. I kept them on ice for 30 minutes, then heat shocked at 42 Celsius for about 25 seconds to let the plasmid enter. After cooling on ice again, I added SOC medium and let the cells recover at 37 Celsius with shaking, and then I plated them on agar with clindamycin. So the next day, only colonies containing the plasmid grow because they have clindamycin resistance. So after colony selection, I needed to confirm that the insulin coding sequence was inserted correctly and without mutations. So I first grew the selected colonies and used the mini prep kit to isolate plasmid DNA. So for sequencing, I chose primers that bind to the T7 promoter and T7 terminator. So the sequencing reaction would read through the entire insert. I then set up the Sanger sequencing reaction, sent the samples for sequencing, and analyzed the result to ensure the insulin CDS matched the reference sequence exactly. So on the left, you see the transformation step. This is where I took my recombinant plasmid and introduced it into E. coli BL21 cells, which are designed for protein expression. And after heat shock and recovery, I plated the cells on clindamycin plates, so only the transformation would grow. Once I had confirmed colonies, I moved on to protein expression. The BL21 cells were grown and induced with induced with IPDG, which activates the T7 promoter to produce large amounts of his-tagged insulin. And on the right, you can see the purification process, harvesting the cells, lysing them, binding the his-tagged insulin to the Ni-NTA resin, and eluting it with imidazole to get purified protein. Thank you very much for listening. Hope you have a great day.